Assalamu alaikum. Welcome to Biology Revisal 2 series. Today's session we are going to be talking about graphical skills and analysis. This is Galuma Miss from Villa International High School, and here with us today
Zafarullah from Ahmadiyya International School.
And Nadine from CHSA.
The main reason why we took graphical skill is that because in paper 3, even in this year January paper, there were around 28 marks related to graph. So that takes around 58% of the whole paper. So it's very important that you know how to handle data related to graph. So what we'll be covering today is tabulation of data, graphical skills in plotting, interpreting from graph, and command words related to it. So let's have a look at it. First, we will have a look at tables. Common error in tables when students are drawing tables. So Zafar sir, can you explain the part related to tables?
Yeah, fine. When you're drawing the tables, better I'll draw one table and explain to you. So the first part, if you can see here, first we draw a box or a table like this. Then the first column will be always your independent variable, the variable that you are varying or the variable that you are changing. That will go on the first column here. For example, if you are varying the temperature, temperature goes here, and you should draw the units here. Don't put any units over here. Let's say you are putting the temperature here, 10, 20, 30, 40, whatever are your values here. Continue there. This is your first column. This one will be your dependent variable. For example, rate of enzyme reaction. So rate of enzyme reaction, but again you have to put the values there, the units, that is per second, okay, or per minute, whatever is that one. And again remember, whenever you are drawing a table, you have to put the raw data as well. Raw data being the trials that you have conducted. You might have done the experiment maybe thrice or maybe 5 times, and you're going to put the trials here. Let's say trial 1, trial 2, trial 3. Let's say you have done it 5 times. Put the values there. So rate of enzyme reaction, you put the values there, and the last column will be the mean, or if you have calculated the standard deviations, you can put the standard deviations here. But here is the mean value. So you are going to put the values here. For example, you have got a value like 1.9, 1.9, 1.9, 1.9. Let's say all are same. And then your mean value will be, you know how to calculate the mean, and you put the value here, whatever mean you are getting here. Okay? And then you continue for the other values as well. So remember, when you are drawing a table, the first column is your independent. Put a heading on top of that one and put the units. Don't forget to put the units there. The next one is your dependent variable and what you have measured here with your units there. Then trials, if you have conducted trials, this is your raw data, and then you are putting the mean value on a separate column there. And don't do any calculations over there, just show the values there. That's how you draw a table there.
Yeah. So for tables also, students should know how to identify the variables. So Nadine, Miss, what are some common errors while they are recording the data in the table?
So some of the common errors that the examiners have highlighted is that putting inappropriate data into the table, and also one of the other common mistakes that the students are doing is that when they are writing down the values in the body of the table, they tend to write it with the units, because when the values are given, when the trial values are given, they are given with units. But students, whenever you are drawing the table, as Zafar sir has shown, only write the unit in the column heading. So units should not appear in the body of the table.
What are all these trials that are done here? Why do we need to do all of these trials?
In biology or in any scientific experiment, trials are actually very important. By doing trials, we can actually increase the reliability of our data. So we can actually show that this is not something that we got once off. Or by chance, yes. And also, if there is any anomalous data in doing the experiment, we can actually remove that from our data set or repeat that to find out if it is actually the true value that we are getting.
So, while they are entering data in the mean, what are some of the errors?
Normally they are not consistent with the significant figures.
Yeah.
So for example, sometimes they put two significant figures, sometimes they put 3 significant figures, and they lose marks in that.
So, what do you recommend, like?
They should be very consistent. Either go for 2 significant figures or they can go for 3 significant figures, but they need to be consistent.
So, consistency is important in writing down the mean. All the mean values should be consistent, whether they are 2 significant or 3
significant figures.
So, shall we go into questions related to drawing tables? Now, this question is related to tensile strength. The breaking force and cross-sectional areas were used to determine the tensile strength of fibers of 5 species of plant, and the results are shown in the graph. So, students are supposed to draw a table from values in a graph. So, Zafar sir?
So here, if you can see the values there, you have these values as from the graph you can take. So as you can read from the graph, you have these values here. So what is there on, first of all, identify the x-axis. So x-axis is fiber there. So already we discussed, the first column will be your independent variable. So here is your independent variable, which is, you can write fiber or you can also write type of fiber. Should I put units there? There are no units for type of fiber. So again, what are the fibers that we have? So we have fibers like curao, then you have jute, you have coir, then you have piassava, all the independent variables, okay? Sisal. So this is your first column, which is your independent variable. You varied the type of fiber, and what did you measure? You measured the tensile strength. The tensile strength. Now already we discussed, at the end of it, put the units, megapascals here. Now, you have to read from the graph that is given there. So I'm just reading out the values here. This is 540, then 250, and then 90, and then you have 130 here. I'm reading from the graph actually. I'm reading the values from the graph, from the y-axis basically. So these are the values that I can read, and I'm going to put it on the second column. There is no other column because I didn't calculate mean or whatever. I'm just putting the values here. These are the values. This is the independent variable. That's how we draw this one.
Thank you, sir. So in the new syllabus, mainly students are asked to read from the graph and draw values from it, isn't it? What are the common errors students do in recording data from the graph?
So whenever you are actually asked to draw a graph, so it has been identified by Zafar sir that on the first column it should always be the independent variable. So being unable to actually identify independent variable is one way in which the students can lose the mark. And another thing is that when entering the data, it's very important to actually correctly read the values from the graph. If you are putting the wrong values, that means that you will not get that mark for the table.
So it's a 3-mark question, right? Yes. Students do not need to know any content knowledge.
Yes.
That's actually one advantage related to it. They can just simply reading from the graph, write down the values without knowing any content knowledge.
Yes.
Actually, that is one advantage of about graphical skills, isn't it? These 21 marks that we are talking about, no need to know the content. They can easily get the marks if they require the graphical skills in how to do it, isn't it? Next, moving on to talking about graphs. Nathan, Miss, can you explain what are some of the errors students do in drawing the graph? A simple graph. Identifying the axes, right?
Okay, so I think it's better if I show it on the table here. Okay, so when you are given to draw a graph, so one of the important things, as we have been highlighting in this lesson, is that been able to identify the independent and the dependent variable. Independent variable is the one that the student or the investigator is actually controlling. So independent, you can remember I control it. So this one always goes on the x-axis of the graph. So this is the x-axis and this is the y-axis of your graph. So your independent variable would always go here. Okay, so your dependent variable, or the one that we are measuring in the investigation, would always go on your y-axis. So these things have already been covered in your lesson, so I'm not going to go into very much detail of this one. So I will just highlight some of the things that you have to keep in mind when you are drawing the graph. So graph points, a graph actually accounts for 4 marks of your paper usually. So the 4 marks are actually given, one for correctly selecting the axis, second one for labeling. So when you are labeling, so you have to give the unit as well. So unit is very important if it is given. Okay, so otherwise if no unit is there, you don't need to write this one. Okay, and another thing that you have to keep in mind is that selecting an appropriate scale. So when you are selecting the scale, students actually tend to, lots of students go to difficult scales where like it's difficult to actually plot your graph. Always it's suggested that when you are plotting the graph, don't use odd numbers. Multiples of 5 is fine, but then even numbers would be easier to plot. Okay, so when selecting the scale again, another thing that you have to remember here is that what is your highest value, what is your lowest value. So this should be in your scale when you are plotting. Otherwise you will, at the end again, it becomes tedious for you to go back and erase and draw it again. Okay, so this is something that you have to consider. One of the reasons why I am saying this is sometimes the students are asked to plot the standard deviation as well. So when they are given the standard deviation, usually you will not have the highest and the lowest value as such. You just have to calculate the highest and the lowest value. So this has to be considered when you are selecting a scale for your graph. Another common mistake students actually do is that whenever they are asked to plot something, okay, so what kind of a graph do they have to plot? Okay, so when plotting, things that you have to remember is that data can be continuous or it can be discontinuous or it can come in discrete groups as well. So if the two sets of data, they are continuous, in this case you will go for a line graph or a scatter diagram. Okay, so if it is a line graph or a scatter diagram, another mistake the students do is that they will sketch the line. So in biology, you are expected to actually use ruled lines, points joined using a ruler. That is very important. And when you are drawing discrete graphs, when the data is discrete, in this case you will go for a bar chart. So when you are drawing a bar chart, in this case you will have to actually make sure that the bars are of equal width and they are also not touching. Okay. In addition to that, when you are plotting, so like I said, when you are plotting, okay, so use a ruler. So here I am using freehand since I don't have a ruler. But then when you are doing it in the exam, make sure that you are joining the points using a ruler here. Okay. And also, do not extrapolate when you are drawing the graph. That is another important thing that you have to think about. So if we have, for example, the origin here, sometimes the students given this data, they will actually extend this to the origin. This will be wrong. Okay, so do not extrapolate the point unless otherwise asked. Okay, so just only join the points that are given in the data set. Okay. Anything else?
Yeah, thank you, Nadine Miss. One thing I notice about drawing of when they are plotting, what Nadine Miss has said it about including points, looking at the scales in drawing the points, right? But when I'm marking the papers, what I have described, some of the points are like so way above beyond the graph point. Now the main reason is that they are not planning well, isn't it? So it's very important that you plan before you start drawing. Having these points into consideration, like standard deviation, things like that. Otherwise, they will go beyond the graph and then plot it near the question itself, isn't it?
That's what I said. So it becomes tedious for the student to actually go back. So it is time consuming. The student don't want to go back. So they
Yeah.
just simply plot something.
Plot it somewhere around it,
Yes.
and then they lose the mark,
lose that
right?
mark,
4 marks, easy 4 marks that they can get for this graph, isn't it? Shall we look at the question? Now, one more thing I want to add. For example, this is just for one set of data, right? If students are given 2 sets of data, how can they identify them?
Yes, that's a very good question to actually highlight, because here in this case, students tend to forget to put a key in the data. So they can label in the graph itself, or it's much easier to have a key. So if you have, like, for example, 2 temperatures for an enzyme reaction, in this case, you would have one line for temperature, let's say 30 degrees Celsius, another line for 45 degrees Celsius. So you would, at the corner of your graph, you can actually have a key stating which line is representing what. So this can be even applied for bar charts as well. So you have to
if there are
distinguish.
paired data,
Yes.
isn't it?
Yeah, again
Paired.
in bar charts they forget to label the individual bars.
Oh yeah.
Yes.
They put the whole overall thingy, but they forget about the individual
Yes.
bars.
Especially if it is a paired data,
Yes,
right?
yes.
For each pair,
They forget to draw
they
that.
forget to label it. And even labeling also, like Nadine Miss have said, they have to give a key for each one.
Yes.
What about shading? We see like students just keep on shading if it is like. So what are the rules that we look at when we are talking about identifying? We say key is given, right? If it is a paired data, how can students do this without like completely shading the whole thing?
They can simply draw a few lines
Yeah.
to distinguish between the bars. So in
Like
your
cross
key, you
lines
have
or
to
straight lines.
straight lines or cross lines.
Because you normally see
Yes.
that the whole thing is shaded, isn't
Yeah,
it?
that is not good.
And it's even time consuming.
Time consuming, yes.
Isn't
They
it?
lose time in that.
Okay, so another thing that normally asked is to calculate the gradient, isn't it? What type of questions do you think they can ask to calculate gradient? It's bio, right? But still they
Yes.
need to know how to do the gradient.
So Galibam is initially also identified in the previous panel also that calculation is very important. So calculation skills are required. So gradient is usually asked for enzyme reactions. So for enzyme initial rate of reaction, we calculate using the gradient. So I hope all of you know how to calculate the gradient. The formula is difference in the values of y divided by difference of the values of the x. By using the right angle triangle that you draw for the
So
those
normally
points.
the lines are not so straight in all the graphs, right? For example, enzyme ones, they normally get a slope like this, isn't
Yes.
it? So how can they do this one?
So if it is a slope, actually in the enzyme reaction, what happens is that as the reaction proceeds, some of the factors become limited. So it means that the rate is going to slightly decrease. So if we were to compare, for example, let's say again temperature, we want to compare the enzymatic activity of 2 different temperatures. So in this case, you have to actually calculate the initial rate. So you can draw a tangent from the initial point and again use the same formula to calculate the gradient.
Thank you, Nadine. Now let's have a look at a question related to drawing graph. The question says a table is given, isn't it, with the data? And then these are the data that they have taken from an investigation. Plot a line graph. They give the type of graph, isn't it?
Yes.
Plot a line graph to show the relationship between detergent concentration and percentage of light transfer. Join the points with straight line. Even that point is given, isn't it?
Yes.
So Nadine Miss, can you explain how they can do this question?
So I will just show it again on the paper. So here, the first point you have to remember. So like I said, points are allocated. So there is 1 mark for axis, there is 1 mark for labeling, there is 1 mark for plotting, and there is 1 mark allocated for the style of the graph. So axis, you have to select the correct axis. So in this case, what is your independent variable? That will be the concentration
concentration of detergent
of detergent
in percentages.
detergent, and labeling should always be complete with units. So here this is given as a percentage. So on your, what we are measuring is the amount of light that is transmitted. Okay, so light transmitted,
Percentage.
this is also again given in percentage. So units are very important. So when you do this one, when you draw up to this, you are actually covering the axis and the labeling points here. Then the next part is to correctly plot it. Okay, so you have to select a scale here in order for you to draw. So style marks will also be considered when you are selecting the scale. So when you draw the graph, make sure that you cover at least 75% of your drawn area. Okay, so graph area should be covered, 75% should be covered, otherwise you will lose this mark for style. Okay, so an appropriate scale need to be taken, and when you are plotting also, you have to consider that even if one point is plotted wrong in your graph, you will not get that mark for the plotting. So it's very, very important. Once you draw, go recheck to see if all your plotted points are plotted correctly. Okay, so when you are drawing the scale, you can actually start with a 0. Okay, it doesn't necessarily mean that every time with every scale you have to start with a 0. Okay, so it depends on the values that are given in the table. Okay, so choose an appropriate scale depending upon the values that are given.
Thank you, Nadine. So graphs are normally for 4 marks, isn't it? So now we just drew a table,
Yes.
3 mark, a graph, 4 mark, 7 marks, isn't it? Only knowing the skill without the content. So students, you should be able to know the basic skills in drawing in order for you to get these marks in. Now next we will have a look at standard deviation. So Zafar sir, can you tell us something about a standard deviation?
So the standard deviations is one of the concepts which many students struggle to answer actually. So standard deviations are nothing but, if you go look at the definition of this one, this is defined as a statistical measure of spread of data, like how the data is spread across the mean. So better I'll take one example and I'll explain on the board here. Now I'm going to explain a little bit about this one here in standard deviations. First of all, let's say I have 2 sets of data. Let's talk about some weight gain protein powder taken by 2 groups of people. For example, from one group A, I got the mean value as 3 kg improvement. Like say the mean of taking the protein powder, the improvement is 3 kilograms. Then I have a group B where I was given the same, where I gave the same protein powder. Here also the mean improvement is 3 kilograms. Now just looking at the mean here is 3 kilograms, mean here is 3 kilograms, improvement I mean. So just based on that I can't decide whether this protein powder is good or this protein powder is good, because the means are same. What scientists or this scientific what should I say, scientific investigations look into is what the spread of data around the mean here. For example, this might be a case where you might have got values like this: 1 kg improvement in one group of people, 2 kg improvement, 3 kg improvement, 4 and 5 kilo improvement. When I do a calculation of mean, this is a mean value that I got. Here I might have got values which are like, for example, I can take values like -3, 0, 3, then +3 and +6. In this case also the mean is 3 here, if you calculate. So mean 3 here, mean 3 here. But here if I look at this one, the spread of data is very, very high. I am getting a value, a person who has eaten this protein powder has decreased his body mass by 3 kilograms, which is not good. If I just look at the mean and conclude from that, I might be wrong, because the data is very much highly spread. You may gain 6 kilograms as well. some people gain 6 kilograms, but there are people who lost 3 kilograms. But this is not consistent. That I'll come to know when I know the spread of the mean, the data across the mean. But here if I just look at the mean, I will not be, it's inconclusive actually. But if I draw this value, the mean is spread across, let's say for example 3 here, maybe plus 2 values and maybe minus 2 values here. This spread is very, very less. So what we do is, when I draw, actually there's a formula for standard deviations, and you are not supposed to learn that, I guess, in your AS. They'll give the formula, I guess.
In A2, yeah.
In A2 they might be asked to calculate the standard deviations. But let's say you need to know the formula as well. So once I do the calculations using the standard deviations, I get a number. So this number I'm going to plot around the mean value. Let's say this is the 3 here, let's say 3 here as well. But let's say I have a minus scale here as well. Here is the minus scale. So this range will be much, much bigger. This is how I draw a standard deviation. So how should I draw? For example, the value that they give, normally you need not calculate it, they give the value here. For example, mean is 3. Let's say standard deviation is given as +1 here, for example. Here, mean is 3, standard deviation given is, let's say 4 here. So how am I going to draw this one? Forget about this one. I'll just better take a new sheet here. Let's say I'm supposed to draw a standard deviation value of 1. So many students ask, well, how can I do this one? First of all, this is your mean value on the y-axis. One is you're going to take +1 value here and -1 here. You're going to draw a bar like this, a vertical bar with hyphen marks here. This is how you draw this one. This is your standard deviation. And what does it imply? What does it show is, it shows the spread of data or the variance of your data across the mean. How much is it spread? And the next thing that you should understand is, what will this give me information about validity or my conclusion? What conclusion does it give me? Means here, if you have a smaller standard deviation, the spread of data is less. That means whenever you are doing repeating the experiment, you are getting consistent values across the mean only. But if I get a larger error bar or a range bar, or I can say a standard deviation here, the spread of data is very, very large. That means it's not reliable. So I'm using the word reliable here. So if you have a smaller standard deviation, the values or this particular data set is highly reliable, and this is less reliable. Then coming on to overlaps. So let's say when there are overlaps between 2 range bars, what does it signify? So where is the overlap here? Here, this value, if I'm looking on the y-axis here, you have an overlap till here. This is all the overlap area here. So whenever you have an overlap, it means there is no significant difference between this value and this value. For example, A and B. Let's say I have so many values here. So between these two, there is no significant difference. What do you mean by significant difference here? Means significance means you are not getting the data by chance. For example, if you have 2 range bars, I'm not drawing the data here. If you have 2 range bars which are not overlapping, I have data set here P and Q here, I can say that my conclusion is there is a significant difference between P and Q. But when there is an overlap, for example, I'll draw one more value here. This is, let's say, S. So there is an overlap here. My conclusion is there is no significant difference between S and Q. What do I mean? Means I am getting values which are overlapping with the Q data. So my conclusion is, just don't say there is no difference. There is no significant difference between S and Q. I hope it helps.
Yeah. So then now we'll go into questions related to standard deviation. The question says that the results in a table were plotted as a bar chart. Complete the bar chart by labeling the x's and showing the standard deviation for fresh juice and for orange juice. Now we have set of data for 6 different types in there. But students are asked to draw only for 2, isn't it? So
yeah, they have been asked to draw for 2, that is for fresh orange and carton orange. So here, if I'm drawing it, Again, the bar graph is already given. I'm not going to draw the whole
Alright.
bar graph here. I'm just going to put the labeled axis. That question is given. Here you're going to say type of fruit. No units there because it's a bar graph here. And then you have here fresh orange, I guess. And then you have the cotton orange. And then you have mean here, mean concentration of vitamin C, milligram per centimeter cube. And then this value is, I guess, 6.8 here. It's already given. It's around 6.8 here, and here the value is 1.3 somewhere here.
Now they are
Now
required to
they
draw.
require you to draw a standard deviation. For 6.8, that is for fresh orange, the standard deviation is 1. Now you already know how to draw this. So what are you going to do? 6.8, it's already drawn there on the bar graph. The length of the bar is 6.8 plus 1, that is 7.8 here. Then minus 1, that will be 5.8. This is how you draw this. And the next one, you have cotton orange, that's 1.3. The standard deviation is 0.2. Maybe somewhere here, you draw it like this. And this is the standard deviation. Now, there is no overlap. There is a significant difference between fresh orange and cotton orange.
Thank you, Zafar sir. That was very well explained, isn't it?
Yes.
Nadine, can you tell us some errors in students when they are drawing standard deviation bar related to labeling of the axes?
So when we have a look at the types of graphs that they are giving in Unit 3, so they focus on completion of the graph. So this is a question where you have to complete the graph. So in this
Labeling.
case, you have to make sure that you label all the axes, units if given, you have to put them there as well. And individual bars have to be labeled. So we've highlighted that in our lesson already. Individual, so you can see that there are many bars in these graphs, so each individual bar have to be labeled separately. And also it's very important here, I think, the question, reading the question.
Yeah.
So it says here, standard deviation values are given for all the types of fruit juices that is given in this graph, but you are asked only to draw for 2 of
Yes,
them.
yes. They may draw for all of them,
Yeah.
right?
So if you draw for all of them, you are
That is
actually using up your time as well.
Again,
You
we
might
talked
not lose
about
the
it.
mark, but then time
Time
is
management,
yes.
because 6 bars are there, and
Yes.
for each bar for them to draw the standard deviation, isn't
Yes.
it? And again, one more thing I always notice is when students are labeling the exercises, they don't write the word mean, isn't it?
Very important. So if mean value is calculated, you have to always, when labeling, state that it is the mean that is represented on the y-axis.
Yes. So that actually concludes about skills in drawing tables, graphs, and interpreting data. Now we will move on to command words related to it, that is analyzing of the data given in tables and graphs. So the first one that we have chosen is describe from the question. The question says, the graph shows the effect of temperature on solubility in water of 3 salts, F, G, and H in the human diet. Describe the effect of temperature on the solubility of these 3 salts.
So coming to the command word describe, this was discussed in the previous panel as well. So as it was highlighted in that panel, in the description part, you don't have to give any justification or reason for why the graph is appearing as it is. So you just simply have to describe the trend that is observed. So for this question, you can see that it's actually a 3-mark question, and there are 3 lines given here as well. So it should be understood that you have to describe each line in order for you to get the 3 marks. So if you look here, you can see that this is showing the effect of temperature on the solubility of 3 salts. So for salt F, you can see that the solubility increases with increase in temperature, while for G, you can see that it increases up to 30 degrees Celsius, but afterwards it is starting to decrease. So if you look at the solubility of salt H, you can see that the solubility is not affected by temperature at all. So you have to mention about all these 3 salts in order for you to get the 3 marks here.
Thank you. Now we'll have a look at explained question. The question says beetroot cells contain a red pigment called betaline, and then they are given a table related to it, isn't it? So it says that detergent permanently damages beetroot cell membrane. Explain what will happen to the rate of loss of betaline if these discs were then transferred to test tubes of distilled water.
Yes, if you look at this question here, you can see this one, the data there, the concentration of detergent as it's increasing, the mean percentage of light transmitted is decreasing. That means it's becoming more and more red in color. That means more pigment is leaking out. So, and immediately if you transfer this disc into a distilled water, but here you can see the distilled water thingy, that means it doesn't have any solute in that, so it will just, the pigment will leak out. If a disc is placed in higher concentrations of detergent, right, that means still that the membrane is permanently damaged, the pigment is leaking out, the betaline pigments are leaking out. But if you look at the last part there, as the concentration of detergent is 2%, the percentage light transmitted is just 50% only. That means it's becoming more and more redder. So if I keep it in distilled water, there is no more pigment left in the beetroot cells to come out. So it may stop or it may be very, very less. But as the concentration of detergent is increasing, the pigment leaking out has increased here, but as I transfer, it will be less because already the pigment has leaked out.
Yeah. Now, as you have seen that we have just described 2 types of command word, describe and explain. Now, that means describe the trends given in it, and as you can see, Zafar explain as to why there was a leakage that is present.
Yeah, they have to give a possible explanation for that.
Yes.
What they described, and they are going to give a valid explanation for that.
Now, in the next question, the same question is they have done with a different type of, in a different concept. In this one, the question says that it's a compare question. In a second investigation, the effect of temperature on the beetroot cell membrane permeability was studied. The graph shows the result of an investigation. Previously, they have done it with detergent, isn't it? Now they want to see the leakage using temperature, how it affects, isn't it?
Yes.
Nadine?
Okay, so students, here it is actually very, very important to actually identify what is being asked in the question. So in the table, in the graph, actually they are showing the percentage of light that is transmitted. So this is an indication of how much pigment has actually leaked out. If more pigment has leaked out, it means that the amount of light that is going to be transmitted through the solution is going to decrease. That is what we are observing from both the graphs. So this command word here, it says compare and contrast. So comparing and contrasting, you have to you can't simply state only similarities for you to get the full marks here. You have to talk about both similarities and differences. So otherwise you will not get the marks. So if we have a look at the similarities between these two, you can see that with detergent also the pigment is leaking out. With temperature also pigment is leaking out. How do we know this? We know this from the graph because the percentage of light that is transmitted is decreasing with each type of treatment. So this is a similarity actually that we observe from both sets of data. But there are certain differences between these two treatments. If you observe the one for temperature, you can see that below 40 degrees Celsius actually the pigment leakage is very, very less. So the amount of light that is transmitted is very, there's a lot of light that is transmitted, approximately around 97% or so light is transmitted. But with detergent we can observe that pigment leakage, it keeps on decreasing. So there is a gradual decrease. There is no certain point at which the decrease is starting, unlike in temperature. And also another thing that we can observe from this graph is that if you look at the final values, you can see with temperature, the final value is around 25%, which is like it indicates that more pigments are there in the solution. More pigments have actually leaked out compared with the different concentrations of detergent. So these are the differences that we observe between the two graphs.
Yeah. About this question is that they have to look at a table and a graph also. So different type
Sets
of
of
ways,
data, yeah.
different sets of data. So detergent is a table, isn't it? The temperature one is a graph. So
yeah.
they should be
Compare
able
both.
to compare both type from a table and a graph. So very important that they know the skills in interpreting this
Yes,
data.
yes.
That's the main reason why we are focusing this session on, isn't it? Next command word that we have chosen is sketch. This is actually very easy.
It's a very,
isn't it,
very easy
Professor?
one, yeah, fine. You have to just label the axis and no need of scale for this one, and then it should be a freehand curve that you're going to draw for this one. It's much easier. Draw the x-axis, draw the y-axis, and plot it with a freehand curve and label that curve. No need to scale, but you have to label the axis as well.
Yeah, that's important.
They have labeled
Even
the
though it
axis.
is a sketch, they still have to
Label the axis. They
label the
forget to do that.
independent and the dependent variable,
Yes.
isn't it? And all the labels should be there, but no strict
Scale and
rules,
label.
right?
Yeah.
In like dotted dot to dot, straight lines.
It should be a freehand. Yeah,
Yes, so it's more like flexible
More
for
flexible.
sketching one, isn't
Yes.
it? Yes, thank you. Now the next one which we are going to have a look at is estimate. So the question says, estimate the percentage of light transmitted at 80 degrees. Use the information in the graph and your own knowledge to support. This is a 3-mark question. You can see there's a lot they are asking you, isn't it?
Yes. So how do we actually estimate a value? So we have to actually evaluate the given data. So if we look at this graph, let's have a look above 60 degrees Celsius. As you can see from the graph, the value is 25%. So when we increase the temperature from 60 degrees Celsius, you are still observing that the percentage of light transmitted is constant. So when we are estimating a value, we have to actually extrapolate from the given data. So if we look at the given data, we are observing that the value is becoming constant.
Yeah.
So we can assume if the temperature is increased above this value, the value, the amount of light that is going to be transmitted is still
There's no
going
line
to
in
be the
80,
same.
isn't it? They have stopped up to 70.
So we have
but
to
they are asking
yes.
to do it for 80, right?
Yes, so we have to extrapolate. So if you draw a line from that one, so it's becoming constant.
They are
because-
asking to do extrapolation.
Yes.
That's
Yes.
the important one, right?
Very important to identify that. And also another thing here is that you have to also give an explanation
Yeah.
why you are suggesting that one. So in this case, above this temperature, we can assume that all of the pigments have actually leaked out. So it means if all pigments have leaked out, the amount of light transmitted is still going to be the same, because number of pigments that is present in the solution is not going to increase with increase in temperature.
Okay. So that actually sums up about graphical skills and analysis. I think we have covered all that we need, that they need to do.
Yes, yes.
Tabulation of data, plotting of data, identifying the variables, and then analyzing from the graph given in there. Isn't it? So that's it on graphical skills and analysis. For our next session, we'll be having a look at level-based question. And then we have got some questions related to some level-based questions which we will be discussing in our next session. So until then, have a good day and stay safe. Thank you.